Welcome to Innovation China's Smart Healthcare Virtual Roadshow. My name is Wang Guan from China Global Television Network, CGTN. As countries and their economies worldwide are struggling to recover from COVID-19, China Association for Science and Technology want to highlight the role of technology in a post-pandemic world. That's why we're here. Innovation China Roadshow is aimed at creating a platform for innovation, entrepreneurship, and nurturing a network of companies, innovators, investors, experts, and talents. The next company we're presenting is NovaScan. This company aims to improve the detection and classification of cancer, be it initial detection, at procedure examination, or in post-procedural diagnosis. It emphasizes improving patient outcomes, increase physician economics, and reduce the cost of care. Now, I would like to invite the representatives of NovaScan to present their company.
My name is Craig Davis, and I'm the CEO of NovaScan. We're an oncology company with a new modality for the detection, treatment, and stratification of cancer. Today, cancer is typically detected through pathology. It's a qualitative art. It takes time. It's hard to do it intraoperatively. It's expensive, and it's especially within medicine. It destroys the tissue by definition, and it works chemically and biologically. We have a new modality that can be used adjunctively, that can complement, that doesn't need to replace, that can be done in seconds and is best done intraoperatively. That's very, very inexpensive. And that's inexpensive in two dimensions. It's inexpensive in terms of physician time, and it's inexpensive in terms of cost of goods of the devices. It's very easy to do. You press a button, you get the answer. Surgeons can learn to do this in minutes, not years. We don't destroy anything, and we work electrically. NovaScan was founded by Dr. Bill Gregory, who was a physicist out of MIT, who spent the last 30 years studying the electrical properties of cells. And 7.5 years ago, he realized something very, very novel. He realized that there's one electrical parameter that relates to how quickly a cell releases a charge, called the Cole frequency, that varies hugely between normal and cancerous tissue. I want to take a look at the graph in here, but I want to take your eye to the horizontal axis. That is a log scale. If that was a linear scale, benign would be on the left and cancer would be somewhere across the street. It is several orders of magnitude. It is really, really huge. And because it is such a big difference, we tend to be very, very accurate when we use it to determine if cancer is present. And NovaScan has a lot of intellectual property around the use of it. This is a platform technology that came out of academia, and work has been done in a number of different areas. It started in mammography, work has been done in breast margin. We have joint ventures in biopsy and in lung, and we have an initiative in skin cancer. But all of these things have several common traits we think are really, really important for cancer diagnostics. The most important thing is it has to work. In 5 clinical trials and more than 400 patients, sensitivity in each and every case is above 98%. And specificity is very, very good too. Secondly, in 2020 and going forward, in the US and in China and around the world, cost is important. These are very low-cost devices. There is a disposable associated with this that can cost in the single-digit US dollars, and the capital equipment in almost all cases is less than 5,000 US. Third, it's simple and fast. You press a button, you get the answer. And we just don't tell you only that there's cancer present. We're also predictive. We inform on that cancer, and we tell you if that cancer is likely to be aggressive or not. The only thing that needs to touch a tissue are electrodes. It can be 0.5 millimeter or smaller. So you can mount them on a needle, or you can use them in minimally invasive surgery. You could use it in targeted drug therapy. Some of the applications are in the doctor's office, some are in the operating room. Our strategy is as follows. The first thing is to partner with large device companies on most types of cancers. I would love to have partnership with some Chinese firms for this. Second one is to develop and build a skin cancer business ourselves. That is not a hospital sale. It's inexpensive to detail. Third is to build out the therapeutics opportunity, because the opportunity in therapeutics is very, very high value. We think we have a pan-cancer solution for cancer detection. We think it can be used several different ways. It can be used diagnostically. It can be used to inform on margin. And we can also tell if it's likely to recur and when it's likely to recur. We are active in non-melanoma skin cancer. We have done lots of work in breast and have JVs there. We have a very active JV in lung cancer. I would love to do work in prostate and colorectal and melanoma, and frankly, every other type of cancer. Our lung products are really, really exciting to us. We have 2 potential products with our JV partner, which is one of the largest medical device companies in the world. The first one is a handheld wand. This can be used, by the surgeon during a lung cancer procedure to tell you immediately, is there cancer present or not? It touches the tip of the wand to that and it will give the answer. The second one involves inducing our electrodes into bronchoscopy to give real-time cancer detection. The bronchoscopy space is changing. It's dynamic. It used to be just rigid and flexible bronchoscopy that go about a third of the way to the lung. Now there are several robotic platforms that can go through the entirety of the lung. An example of one of these is the RS platform. J&J acquired that for $3.4 billion US upfront when it was pre-revenue. For products like this, NovaScan greatly enhances the value proposition that they provide. There are a couple of unmet needs today in bronchoscopy. One of them is that lesions that are not visible under imaging are typically not able to see. The bronchoscopy won't help you if you don't see them. So you only see what's in the airway with bronchoscopy. The other one is when you go with bronchoscopy and you go to the lesion and you take a biopsy, those lesions are heterogeneous. So you might have a cancerous lesion, but you're only taking non-cancerous material, will give you a bad answer. And the Roche processes that used in real-time assessment have poor specificity. Our electrodes can be employed in robotic and normal bronchoscopy. For a handheld surgical tool, we answer several fundamental questions. First, is the margin clear during a procedure? Are the lymph nodes involved? And if so, which ones? Or is this tumor or is this normal tissue? This has lots of value proposition, lots of value in lung cancer, but frankly, it has value in many other types of cancer surgeries. So with our partner, they came and they approached us, they asked us to work, and they asked us to demonstrate that the cold frequency can be used to detect cancer in lung tissue and lymph nodes. At that time, we had no data in lung. So we conducted a trial with them and that they paid for. They set a pretty high bar, and I'm happy to say that we exceeded it. So now we are building both a bronchoscopy project and a handheld wand. These products will get FDA clearance in 2022 and 2023. This is non-dilutive capital to NovaScan. It comes in to build this, and we will monetize at the end. We also have a partnership in breast biopsy. Today in breast biopsy, when it's taken, the results are sent to a central lab, and it can take a week to 10 days before they come back to the answer. It depends upon the hospital system. There would be a value proposition in knowing that immediately. Our technology allows them to do that. We have several different value propositions. The first one is we eliminate the patient angst associated with waiting for that time. We can tell you immediately if there's cancer present or not. The second one is that we can help pathologists differentiate between early stage cancer and atypia. Pathology is very, very good at determining normal tissue, and it's very, very good at determining advanced cancer. When you're doing breast biopsies, the amount of tissue you have is very, very small. And mistakes are made about 30% of the time between atypia and early stage cancer. We can help inform on that. The other one is we can inform on cancer aggressiveness at the point of biopsy. It's akin to a genetic test that can be done point of care. Genetic tests are much more expensive than the disposables for biopsies. We think we changed the value proposition there. We have done a study in this space, and I'm happy to report our results were perfect. Every single time there was cancer, we said there was, and every time there wasn't, we also said that. So we can effectively inform on the presence of cancer in real time, and we may be able to help differentiate between atypia and DCIS. The workflow here is simple. You take the biopsies, you put them in our disposable, you put it in the cartridge, press the button, and the results come out immediately on the screen. This product will launch at the end of 2021 or early 2022. We're doing a skin solution ourselves, but this is actually broader than skin. It can be used for almost every ex vivo assessment. But for skin cancer today, when you try to get rid of it, there's two different ways to treat it surgically. The most common way is you just cut it out. And because there is no intraoperative assessment of margin during the procedure, the surgeon makes a large incision, and it is disfiguring. And because you did not assess margin during the procedure, it can come back. The other modality for doing this is something called Mohs surgery. In this case, the surgeon tries to excise only the cancer. Think of something on your eyelid or something like that. So you're assessing only the cancer, and then you do a frozen section assessment while the patient stays in the chair. So you freeze the tissue, you stain it, you interpret it. Where you have cancer in the margin, you come back and you take more. Then you freeze and you stain and you interpret it, and you continue this until you finish it. This is the gold standard. But there are a couple of problems. First is because this freezing, staining, interpreting process is long, it can take 40 minutes or more, and there is no way to know whether it's going to be 1 time or 2 times or 3 times or 4 times or more do it. So it's very, very inefficient to schedule. Using our device and our technology, we combine them both. So you do a tissue-sparing surgery just like in Mohs, but instead of having to do this freezing, staining, interpreting process with a central lab or with a lab on the premise, you just place it on a device and we scan it and we tell you immediately if there is cancer in it. Our initial trial was 98% sensitivity and specificity. We published it in the Journal of the American Academy of Dermatology, which is the top journal in the world in dermatology. We look for FDA approval late next year. I can show you a demonstration of our device. So this is our prototype device. This is not the commercial device, but it is where we today, and you get a handle on how this works. See our device. It's next to a computer, so you get an idea on how big it will be. The commercial device will be much smaller. That is a disposable electrode that's placed on the device. That is what touches the excision. Beneath that electrode, there are pogo pins, and that's how we scan across the entirety of the excision, and we can tell you if there is cancer in the margin, but not only if, we tell you where, so you know where to take more. So the surgeon would place the excision on the electrodes. He or she locates that. Then he communicates with the laptop to tell her where to scan. So she's, on the graphical user interface is telling you which electrodes to scan. And now he scans. Everywhere that there's no cancer in the margin, it's white. If there is cancer in the margin someplace, it'll show up as red. And you see there's a couple of different locations. So simple and straightforward and very, very fast. That is our device, and that is our product concept for a commercial device. You see how big it is by comparison to the cell phone. There are more cases of non-melanoma skin cancer than there are all the other cancers in the world combined. In the United States, standard excisions are about 30% of the market, and Mohs are about a quarter of the market. We see ourselves replacing standard excisions and making big inroads into Mohs. If you extrapolate this market globally, it's about $1 billion for us, and it can be focused. It's a focused sale, and it can be serviced by a small sales force in the U.S. We would partner in countries like China and others. We have done the prototype device that you have seen. We're working on detailed design of our commercial device. We look for FDA clearance late next year. The final area in which we're active is in targeted drug therapy. Our electrodes are so small, you can put them on the tip of an injecting needle when you're using for non-surgically treated cancers. Think of a cancer that could be in the liver near the bile duct or something where you cannot surgically resect it. So often those are treated by dosing directly into the tumor, but there are under imaging, under CT, but there are challenges with that. Imaging is not perfect. Imaging is good, but not super. So the interventional radiologist doing this, may be adjacent to the tumor, but not directly into it when they're injecting. And that has impacts. It translates to poor efficacy. It translates to side effects. By taking our electrodes and putting them directly on the injecting needle, we can solve that problem. I'm happy to say that Dr. Charles Link, who is the founder of New Link Genetics, has joined us and is leading this effort. Dr. Link is a noted medical oncologist, and New Link Genetics, he founded it in his living room, and he took it public, and I think at the market cap at the height was about $3.4 billion. Dr. Link is also the developer of the Ebola vaccine that is sold by Merck. Our leadership team has accomplished, over the course of our careers, we've had multiple exits. We've raised several $100 million, and we've taken 6 products from concept to full regulatory clearance, all Class 2 and Class 3 FDA products. The board includes one of the founders of Hologic, myself, the chairman, the person that was in charge of IP for GE Healthcare, and Dr. Link. The scientific advisory board I'm very proud of. They are all luminary surgeons, presidents of each and every important society in their respective specialties. Our projections for US skin, and this is only US skin. This is nothing in China. This is nothing in the lung. This is nothing in breast biopsy, nothing in targeted drug delivery. So they're all additive to this. But you're looking for about $5 million to get the FDA clearance. We look to do that late next year. So in summary, we think we have a great technology with really, really good data. It stands by itself and it's published in top journals. The products are, in all cases, very, very inexpensive, fast, and easy to use. Our electrodes that are disposable per procedure, they're cost in the single-digit US dollars. And the capital equipment in all cases is less than 5,000 US dollars, in many cases less than 1,000 US dollars. We have lots of different IP. The initial IP is on the use of the cold frequency, and in each and every application, we tend up filing more IP, and it's filed around the globe. We think, depending upon how you're using this, we can benefit every constituent in the space, be it the physician, the patient, or the payer. We have struck deals with some of the largest companies in the world in several different areas. We're looking for more. I would love to strike a deal with a Chinese firm. And we're very, very capital efficient. We think there's the potential for multiple exits here. Thank you very, very much. I'm happy to take any questions.
Our experts to comment on this project is Li Xiaoqiang, founder and chairman of Your Health and Wellness International Medical Clinic.
Hi, Chris.
Hello.
Yeah, hi. Wonderful. To begin with, I think that this is a very interesting value proposition from your product. Assuming that the cold radio frequency scanning can tell, like your graph said, that can tell the difference, significantly can tell the difference between benign tissue and malignant tissue. This would be a wonderful product and a great potential. To begin with, I think that immediately to tell if this is malignant or not, it can be used to replace the frozen section OR that is widely used currently to quickly tell the difference between malignancy or benign for the surgeon to decide whether this is going to be extensive incision or this is going to be a conservative incision. That is part of that. On the other hand, I think the more wider use of it would be to use it to detect very quickly the superficial cancer, whether this is a skin cancer or the cancer on the surface of the GI tract or the bronchus. Depending on if your sensor can be mounted into the endoscope, whether this is a broncho endoscope or this is a GI endoscope, that there is a widely potential for application. But this was a great idea. Well, assuming that you can actually engineer from engineering standpoint and also from true application standpoint, you can actually materialize the true sensitivity and specificity of the criteria that you proposed in your presentation. On the other hand, the clinical setting is much more complicated. For instance, I don't know that. Well, this is the first time I've heard from the cold frequency, radio frequency session health difference without digging into much of the physical chemistry or physics of the radio frequency to tell the difference between cancer, benign tissue and malignant tissue. In clinical settings, it varies a lot. For instance, if it's skin cancer, there is squamous cancer, there is melanoma, there are other different types of things, and also the size can be very different. The depth of the tumor can be very different depending if this is going to be above the basal membrane or below that, whether this is invasive or not. Without knowing the exact mechanism of the detection, it's very difficult to extrapolate to try to understand what is the potential clinical effect.
I'm happy to show you and sort of explain the method of action.
Sure. That would be very important because eventually all this clinical application really eventually boils down to the core, the mechanism technology, whether this is a really specific and sensitive or not. I mean, 90% sensitivity is wonderful. It's great enough, and then 80% of specificity is also very good. Obviously, for treatment, you wanted to have probably higher, to guide for treatment, you want a higher specificity. However, for screening purpose, a high sensitivity will be good enough. So there is a wide potential application point that you can develop, and also the engineering part of that will be what it is you can minimize the volume of this sensor to make it into a various complicated, sophisticated medical instruments to detect the tumor in various parts of the body. But generally speaking, I think that the technology is very interesting and it gives a lot of imagination for both investors and potential users. Thank you very much for that.
Thank you.